Professor. It's my pleasure to present our work to International Research Awards on Oncology and Cancer Research. My name is Jian Zhang, and I'm from Guangzhou Medical University. Today, the presentation title is Identification of Necropotosis-related signature and tumor microenvironment infiltration characteristics in non-adenocarcinoma. Lung cancer is a leading cause of cancer-related deaths in China and globally, and lung adenocarcinoma is one of the main types of lung cancer, which accounts for nearly 40%. And over the last decade, treatment strategies like radiotherapy, molecular target therapy, and immune checkpoint inhibitors have greatly advanced. However, the outcome of non-adenocarcinoma remain unsatisfactory, and the 5-year overall survival rate is nearly 80%. Thus, defining novel prognostic prediction method for lung adenocarcinoma in order to find new strategies, targets, and improve patient survival still need to be further employed. And in cancer, apoptosis resistance is one of the most common hallmarks, making it difficult to develop new strategies for fighting cancer. And the activation of cancer-related of growth signals, metabolic reprogramming, and dysregulation of tumor microenvironment contribute to cancer cell resistance to death, allowing it to proliferate or metastasis uncontrollably. Thus, inducing cancer cell death has emerged as a promising research direction in the field of cancer treatment. And necropotosis is currently one of the most common programmed cell deaths, activated by the receptor interacting protein kinase 1 and 3, mixed lineage kinase dome-like protein, and their targets. And necropotosis have been reported to be associated with several human diseases, including inflammatory disease, infectious disease, neurodegenerative disease, and cancer. However, necropotosis plays a double-edged role in the tumor microenvironment. On the one hand, necropotosis episodes may trigger a strong adaptive immune response, which may prevent cancer progression. And on the other hand, the activated inflammatory process may aggravate cancer tumorigenesis. Thus, the objective of this work has three aspects. The first is to assess the necropotosis-related signature and analysis the correlation between the necropotosis and the tumor microenvironment characteristics. The second, to construct a necrosis score and evaluate necropotosis pattern of each patient. The third, to evaluate correlation of necrosis score with the immune checkpoint expression, immune checkpoint inhibitor response, and clinical benefits. And this is the workflow of this study. Gene expression data and survival data are downloaded from the TCGA database. A total of 14 necropotosis regulators are identified from public studies. We first performed unsupervised clustering, and 3 modification patterns, cluster A, cluster B, and cluster C, were identified. We first performed function analysis like GO and KGG and GSVA analysis among 3 modification patterns, and then evaluate of tumor microenvironment cell infiltration, and we correlated with the nuclear proteases modification patterns and tumor microenvironment cell infiltration and construct immune phenotype, immune excluded, immune inflamed, and immune desert. And we then performed differential expression analysis and identified nucleoproteases phenotype related genes and further performed clustering, and 3 nucleoproteases gene cluster were identified, like nucleoproteases cluster A, cluster B, and cluster C. Similarly, we performed functional analysis and survival analysis. Next, we perform univariate Cox regression and PCA analysis and construct a nucleoproteases gene signature we named nucleotide score, and a geo metacode to validate the nucleotide score and the mutation status of driver genes between low and high nucleotide score. At last, we performed survival analysis, correlation nucleotide score with TMB, with PD-L1 and tumor microenvironment infiltration. And last, in immune court, we further evaluate infiltration of immune cells and tumor cells, immune phenotype between low and high nucleotide score. Last, response to immune checkpoint therapy and prediction progenesis of immune checkpoint inhibitor therapy. And here is the results. A total of 14 regulators were identified, and among 561 samples, a total of 52 samples showed mutations of nucleoproteases regulators with a refrequency 9%. As shown in this figure, 10 nucleoproteases regulators like EZH2, RIPK1 to RIPK3, which exhibit the same mutation frequency with the frequency 1%. However, there were 4 regulators which didn't have any mutations in non-adenocarcinomas, like BIRC2, USP22, and PGM5, HMGB1 with the frequency 0%. And the analysis of copy number alteration analysis indicates that 11 nucleoproteases regulators displayed different extents of amplification of copy number, and the PGM5, MLKL, and HMGB1, which showed in most of cancer showed copy number loss, and other regulators showed the copy number gain. And the location analysis shows that the location of CNV of 14 regulators on 23 chromosomes, and the location were scattered and unorganized. Further expression analysis shows that the 7 nucleoproteases regulators were upregulated, and while NDRG2, which is downregulated in tumor samples. And these results indicate that the complexity of genetic alteration and impression in nucleoproteases of regulators displayed a crucial role in the tumor genesis of non-adenocarcinoma. And to discover the roles of nucleoproteases regulators in progenesis, we first perform a univariate Cox regression analysis, and to further explore the interaction rules of among the regulators with spearman correlation analysis also performed. And as shown in this figure, we find that the comprehensive landscape of nuclear cis-regulator interactions, regulation interaction, and progelastic interaction in lung cancer. We found that on the significant negative correlation was found, like CYLD and PGM5, CYLD and TRAF2, and NDRH2, TRAF2. And on the other hand, the most interaction of nuclear proteases regulators show significant positive correlations, like MIKL and CSP8, MILKL, and BRC3. To further improve the rules of nuclear proteases-related patterns in the TME, the patients with different nuclear proteases patterns were classified by unsupervised clustering. The three distinct nuclear proteases modification patterns were identified based on the expression of 14 nuclear proteases related regulators. And the survival analysis shows that the three nuclear proteases modification patterns has a distinct survival difference. The nuclear protease cluster A has a better has a notable disadvantage compared to the other clusters. And these results indicate that the crosstalk among the nuclear proteases regulators may mediate the construction of the nuclear proteases. And the immune cell infiltration analysis also found that the immune cell amongst three modification patterns were completely different. These results also indicate that the nuclear protease modification patterns were involved in the regulation of immune cell infiltration. And to further verify the potential regulation mechanism, unsupervised cluster were further performed to describe the genomic subtypes based on the 33 nuclear processes phenotype with different expression genes, and consistent with the results of nuclear cis patterns. The results exhibit that three distinct gene clusters were identified. And the survival analysis also found that the nuclear proteases gene cluster C has a better survival compared with the other gene cluster, and the gene cluster B has the poor survival. And the two patient nuclear and the nuclear score were further to quantify the nuclear proteases patterns of individual patients with lung adenocarcinoma. And the as shown in figure 3b, the nuclear proteases gene cluster C has a high nucleotide score. Among nuclear proteases modification patterns, the modified nuclear proteases modification pattern C has a lowest neurocortex score, and these results indicate that an elevated nuclear score correlated notably with the immune inactivation, and the nuclear score could be used to identify the immune nuclear proteases patterns and TME characteristics in the lung adenocarcinoma. And to further verify the value of the nuclear score, pattern in the TCGA GU were divided into high and low nuclear score groups, and the survival analysis shows that a low nuclear score exhibits a lower survival consequence. And the survival analysis of GU mitochondria also identified that patients with a high nuclear score was improving outcome. And the further GSE 13123 court identified that a low nuclear score was associated with a poor overall survival. Besides, TCGA court with nuclear score indicates there is a significant immune checkpoint expression, including PD-L1 and CLL4, than the high nuclear score subgroup. This results indicate that the crosstalk between the nuclear score and the immune checkpoint in TCGA court was further employed. The high nuclear score and the high PD-L1 or nuclear score and CTLA4 was analysis. We found that the high nuclear score and high CTLA4 or high PD-L1 had a greater survival than low nuclear score and low PD-L1 or low PD-L4 subgroup. And too low, the practitioner rules of the nuclear score in immune checkpoint inhibitor. We investigate whether the nuclear score could predict clinical response to PD-L1. As shown in the figure 7a, the low nuclear score had an improved clinical response to PD-L1 immunotherapy. And projected survival analysis also shows that low nuclear score present a notable clinical benefits, and these results indicate that nuclear proteases patterns are significant associated with tumor microenvironment characteristics and immune phenotype, and that nuclear score could act as a promising biomarker to forecast the response to immune checkpoint therapy. Based on the results above, we conduct conclusions. Nuclear proteases-related regulators were involved in regulating the complexity of tumor microenvironment characteristics. Target the nuclear proteases-related regulators may guide the clinical immune checkpoint inhibitor therapy. And acknowledgements. Thank you for this author's contribution to this work. Thanks the Guangzhou Medical University and affiliate Cancer Hospital and the Institute of Guangzhou Medical University. Thanks for the funding of National Nature Science Foundation of China. And this is the article. Below is the website you can find. And thanks for your listening.